Hi, I'm William Liang, and I recently presented this paper by Ioannidis in 2005, Why Most Published Research Findings Are False, at the U Chicago Reproducibility Journal Club. So I figured I'd record for those of the people who missed that meeting or who are interested in this paper. Firstly, briefly, a quick shout out to reproducibility. What is reproducibility? It's a journal club initiative for early career researchers hoping to build a community that is engaged in improving research practices. And the papers that are discussed at a reproducibility journal club are usually relevant to the replication crisis and looking at improving science. Mainly those papers that focus on open and reproducible research. So onto the paper. It's a very provocative title by Ioannidis, Why Most Published Research Findings Are False. So let's unpack what evidences the claim that less than 50% of published findings are true, and why does something like this motivate open science and reproducibility practices. So let's start an example. Let's start off with an example with p-values. So a p-value is defined as the probability of a sampling result if the null hypothesis was assumed to be true. Let's say we obtained a p-value of 0.01. We adopt an alpha of 0.05, so we make the decision to reject the null, and we conclude that there was a statistically significant difference. So is the following statement true or false? The probability that we made an incorrect conclusion is equal to the observed p-value, 0.01. Give you a moment to think about that statement. It is false. So an observed p-value does not equal the false discovery rate. So even though we observe this 0.01 p-value, it does not mean that the chance that the decision we make when we reject the null is false is also equal to that 0.01 value. We can change the framing a little to make it a little bit clearer. So using the inverse framing, was the probability that we found a true effect equal to the observed p-value 0.01? So because we observed this p-value of 0.01, is the 0.01 the probability that we found something true? And the answer is no. So we can demonstrate this. We can look at this. So what we're talking about here is known as or referred to as the positive predictive value, or PPV. And this is what we're really interested in as scientists. What is the probability that an observed significant p-value indicates a true effect? So what is the probability of a true effect given we observe the significant p-value? So an example of calculating the PPV here, I'm adapting an example from a textbook by Richard McElreath called Statistical Rethinking. I don't think he endorses this particular example to demonstrate this point, but I will show it anyway. Let's say when a hypothesis is true, our probability of a significant result is 0.95. This is referred to or typically defined as our power. So our probability of finding a significant result given a true effect is 0.95. And let's say when a hypothesis is false, our probability of a significant result is 0.05. This is our false positive rate, sometimes referred to as our alpha. So the probability of a significant result when there is no effect, so given no effect, is equal to 0.05. And let's just say that across all hypotheses, 1 in 100 are true, so the probability of a true effect is equal to 0.01. So using these values, we can actually calculate the PPV. So take, for example, the scenario where we do have a true effect, so that's 0.01% of the time. Our ability to detect that true effect is our power, which is 0.95. So the significant p-value that we observe will happen 0.01 times 0.95% of the time. So that will give us 0.0095. Sometimes we have a true effect. We don't observe the significant p-value. So that happens some of the time, so 0.01 times 0.05. There's also the scenario where there is no effect, right? Where so in our scenario, in our hypothetical here, we've assigned this a probability of 0.99, right? 1 minus the probability of true effect. So we have a alpha of 0.05, a false discovery rate of 0.05. So in this scenario, which is 0.99% of the time, so 0.99 of the time, 99% of the time, we observe a significant p-value 0.05%, 0.05 of the time, 5% of the time. So we can observe a significant p-value 0.99 times 0.05, which gives us something close to 0.05. Most of the time when there's no effect, which is true, like most of the time when in this scenario when there is no effect, we won't observe a significant p-value. So this happens most often. You can see here, no effect, not significant, 0.99 times 0.95 is 0.9405. Now, we're only interested in when we observe a significant p-value, and if you take those two scenarios only, you can see here the significant p-value can occur 0.0095 plus 0.0495 of the time. So what was the probability that it was a true effect? The significant p-value we got came from a true effect, given it was a significant result. Well, we take that initial top probability here of 0.0095 and divide by the probability of obtaining a significant p-value. So 0.0095 divided by 0.0095 plus 0.0495, that gives us a value of 0.161. So what that means is the probability of a true effect given a significant result in this hypothetical is 16%, is 0.161. And so that's very low, right? We hope the majority of the time we see a significant result, it indicates a true effect. But in this scenario, in this hypothetical, it is only 16% of the time. And so what hasn't been mentioned here, and which John Ioannidis in his paper calculates and accounts for, is that lots of findings are pushed from being false or from being non-significant to significant, right? So there's some bias in our published findings. And this could be a result of p-hacking, which is when you increase your sample size or to try and find a significant effect, or p-hawking, which is looking for when you have significant p-values to say what your hypotheses were, and erroneous statistics and other things that may push not significant results into significant results. So this is referred to as bias here, and in this figure, in this graph, you can see here there's 4 lines indicating bias here, where U equals 0.05, meaning a low bias, and in yellow, U equals 0.80, meaning a high bias. So in this figure taken from the Ioannidis paper, here I've cut out the specific panel for power equals 80%. So that is, we have the chance of us finding a significant effect when there is one is 80%. And you can see here, as down the bottom, the pre-study odds, that's equivalent in the hypothetical example that I presented, the probability of a true effect. Now, if it's very unlikely to be greater than 1. We don't know what this actual number is. We don't have an estimate for this number. But as you can see, for us to have a PPV higher than 50% at 80% power, our pre-study odds tend to have to be very high, have to be closer to 0.4, 0.5. In the scenario where it's low bias, when it's blue, you can see that it reaches 50% earlier. Of course, by no means we don't want 50% to be the standard. We want it to be closer to 100%. We want our significant effects to indicate true effects rather than false effects. So, but you can see here, as the bias gets higher and higher, the PPV calculations get lower and lower. And so we know some bias exists. We don't know how much, and we don't know which of these values is the true bias value, but you can see here that it's difficult to obtain a 50% PPV. So this is in the high power scenario when it's 80% power. If you also look at 50% power, then it becomes much more difficult to obtain a PPV of greater than 50%. And at 20% power, so a very underpowered study, you can see here that you need to have very high pre-study odds to be able to get to a PPV value of 50%. So yeah, when is a PPV greater than 50% achieved? Well, you can see here on the 3 columns on the left-hand side, on the 1 minus beta is your power here. R is the pre-study odds, so the chance of probability of a true effect. And U here is the bias. And you can see here, there's only 2 scenarios where a 50% PPV is achieved based on these values. An adequately powered RCT with little bias and 1-to-1 pre-study odds has a high PPV, or a confirmatory meta-analysis of good quality RCTs, which is to say that because it's confirmatory and you've seen previous results where you got significant true effects, then you're likely to have a high PPV. But all these other examples show a PPV less than 0.5, right? Less than 50%. For example, an underpowered but well-performed RCT will still only achieve a PPV of 0.23, for example. So here are some corollaries that Ioannidis points out. Firstly, that smaller sample sizes mean less power, which lowers the PPV. Smaller effect sizes means less power, which also lowers the PPV. And exploratory research with larger number of relationships has lower PPV than confirmatory research. And these 3 here, the last 3 that he pointed out, I think are the most important. The greater the analytical flexibility, which is the greater that the chance that people can turn negative into positive results, this lowers the overall PPV for the field. And this could come from many sources. This could be from researcher degrees of freedom to p-hacking and so on and so forth. So this is referring to the bias here, where scientists may turn negative into positive results. The greater the conflicts of interest to produce positive results, the lower the PPV. So this is, for example, if you have a conflict of interest in funding, so maybe you're a pharmaceutical company and you want to show your drug works, it's in your interest to find a positive result for your drug. So you might push your negative results into a positive result. So the greater the conflicts of interest in any field, the lower the PPV. And the PPV decreases with more scientific teams involved, especially if there's a trend of alternating positive results followed by negative results refuting claims. So this occurs in the literature some of the time when a lab produces a nice shiny positive result and they put it out, but then another lab disproves it or tries to disprove it or is unable to replicate it or shows that the result isn't true. And then the original lab will then refute that claim by confirming that their result is significant and is positive and is true, and it goes back and forth and forth, back and forth. And that overall interchange decreases the PPV of that field as well. So are most published research findings false? Well, so the PPV of 50% and above does appear to be difficult to obtain, right? How many studies in science are adequately powered, well-designed, randomly controlled trial, researching an effect that is likely to be true? This cannot be said to be the case for all scientific studies. And we often fail to reproduce the results of others. So in the reproducibility project psychology, 36% of significant results were replicated. So that's less than 50%. So often we don't replicate the results of others. So So does that mean are we in a reproducibility crisis? Well, according to Finelli 2018, no. For most findings in psychology and neuroscience to be false, the true hypothesis rate would have to be much lower than 50%. We don't have an idea what this actual value is, but it's what this might refer to, or what this might entail, is that we have good theories. What's not accounted for is that we have theories that inform our experimental design. So scientists don't just randomly look in the dark. We have logic and models that will determine what is the case. So perhaps the true hypothesis rate is not much lower than 50%. And while there is some bias in the literature, while there is scientific misconduct and questionable research practices, they appear to be relatively small in the literature, about 1 to 2%, and they don't seem to be distorting the greater scientific literature. And there have been times where we have replicated results. So a many labs project did replicate 10 of 13 studies across various experimental settings. So we aren't in a total crisis. But the truth is we can do better science. So scientists are newly assessing the reproducibility of our research, right? This is made possible with powerful information and communication technologies. We're able to send and share data for the first time and look at certain values and certain aspects of our research. But we should also definitely better power our evidence and diminish our bias and understand what exactly our pre-study odds are when we think about our experiments and our experimental design. Perhaps instead of calling it a crisis, the best thing we can do is actually think about this as a revolution of doing better science with transparency and reproducibility in research, hopefully with initiatives like this one, like reproducibility. Okay, so if you enjoyed that little video, that's my Twitter, you can follow me there, and I hope that was useful.